lovely out there? Do you remember a couple weeks ago?
No.
We sang this thing. I thought we might just go through it again. What did we say? Oh, the Oregon weather's dowdy, 'cause the sky... No. You know, your hearts aren't in it. I can tell. You gotta feel for these things, okay? So I thought instead we might try this. Same tune. So the forecast says much more raining. It's of this that I'm complaining. I get cranky from all the slop. So make it stop, make it stop, make it stop. It is dark and it's gray and gloomy. Oh, the climate's out to screw me. I'll go crazy from drip, drop, drop. Please make it stop, make it stop, make it stop. Every time that I look outside and espy the sun, then I know when our weather is Jekyll Hyde. It's solely to make a rainbow. Well, now I know when the rain is dropping that my clothing will be sopping. I don't care if it's good for crops. Just make it stop, make it stop, make it stop. Your heart was much more in that one, I can tell. Yeah. Okay. Back to something less fun. All right, so we got started on translation last time, and today I want to wade through as much of that as I can. And we talked about the genetic code, and we talked about transfer RNAs. Today I'm going to go through and talk about some of the nuts and bolts of translation, sort of how the process occurs at the sort of molecular level. So that's what is on tap for today. Now, I showed you last time the transfer RNA, and what I didn't show you was its actual sort of projected three-dimensional shape. When we draw transfer RNAs in 2 dimensions, we typically draw them as a cloverleaf structure because we're drawing to show the individual base pairs that form in the self-complementary structure. But if we look at it in 3D, this is a sort of 2D projection of the way it actually looks. It has sort of an L shape. It looks kind of like that, where the anti-codon loop is down here at the bottom, and the CCA terminus is up here at the top. And that's, I think, useful to see. Obviously, you're not going to draw something like that, but you get a different feel for this guy after you see that. Now, it's at the CCA, of course, where the amino acid gets transferred or covalently attached in an ester bond. And I want to spend the first part of the lecture talking about that attachment. Okay? So the attachment of the amino acid to the terminus, the 3' terminus of the transfer RNA, is a critical step. Okay? Without the proper attachment of the amino acid to the right tRNA, the genetic code would have no meaning. All right? So it's essential that we get the right amino acid up here onto this 3' terminus. Okay? Very, very important thing. We have to get the proper amino acid out there. Well, where is the information for what to put up here? Well, it turns out it's down here. Okay? Now, that was kind of an interesting problem when people first discovered it. Before they realized this guy had an L shape, they knew the dimensions of the enzymes that put the thing up here. They knew how far apart this would be from this if it was that straight cloverleaf shape that we saw. And they realized that the enzymes weren't big enough to span all the way from here down to here. So when they found the L shape, where this guy was actually bent down and closer to this, they realized that this L shape had an important function, and that was that it allowed the enzymes to be able to read this and put the proper tRNA on there. So that was kind of a cool thing that we learned by studying the structure of a molecule. Well, the enzymes that have to put the amino acid on there have a name. They're a class of enzymes, and they're called aminoacyl tRNA synthetases. Big mouthful of a name. Aminoacyl tRNA synthetases. Okay? Why don't we call them ATRSs? Okay? Aminoacyl tRNA synthetases. Yes, sir? The bent structure is the L structure. That's right.
What's the more stretched out structure?
Well, there is no stretched out structure. If there were a stretched out structure, it would just be flat. It doesn't have a name. Yeah. Okay. All right. So these, it turns out that there are 20 aminoacyl tRNA synthetases. And not coincidentally, there are 20 amino acids that make it into proteins. You would be tempted to say there's one aminoacyl tRNA synthetase for every amino acid, and that temptation would be correct. Okay? So there's one aminoacyl tRNA synthetase for each amino acid. That means that each aminoacyl tRNA synthetase has to be able to recognize all of the tRNAs that a given amino acid will be put onto. Remember that we have different anti-codons. Some of them have 4, some of them even have 6 anti-codons. So the aminoacyl tRNA synthetase has to be able to recognize all of those guys. The individual enzymes have names, and to be honest, I don't spend much time on the names, but I'm sure I just mentioned that because if you look at this guy, 3-anyl tRNA synthetase, that obviously is the aminoacyl tRNA synthetase that puts threonine onto the proper tRNAs. Okay. This reaction that puts the amino acid on there is an energy-requiring reaction. You can actually see the reaction up here. And you will notice that in the process, we form this bond, amine-boacyl. I got an extra B in there. Okay. Amine-boacyl. That aminoacyl bond that is linked between the tRNA and the amino acid. Okay? Blah, blah, blah. We don't need that. Now, when we talked about DNA polymerases, we talked about the fact that they have a proofreading function. And although I didn't mention it for RNA polymerases, it turns out they have a proofreading function as well, but it's not built into their enzyme. They use actually a separate protein to do some proofreading for transcription. Well, all of these processes are important to read properly because if we misread to any significant extent, then we're not going to make the proper proteins that we want. And of course, if we can't make the proper proteins, then the cell may be hosed. It turns out that the aminoacyl tRNA synthetases have proofreading sites as well. Okay? They have proofreading sites as well. The proofreading that they do is not as rigorous as the proofreading that is done in DNA replication, for example. Okay? Not as rigorous. I'll show you a table in a second showing that it's not practical, to be as rigorous as it is for DNA replication. Okay? And this is simply showing that we have a place where the amino acid can be put on and a place where that amino acid can be evaluated, essentially. Okay? And so that evaluation, we can think of the editing site as the proofreading site for the aminoacyl tRNA synthetase. If the wrong amino acid gets put on, it gets clipped off. Okay? If the wrong amino acid gets put on, it gets clipped off. Now, when we think about the accuracy of putting in amino acids onto tRNAs, we have to think of what's the cost of that, okay, for what we're getting. If we make an error in DNA, that error is there forever. If we make an error in making RNA, that error is not there forever. Remember, we recycle, we break down our RNAs. Okay? Similarly, if we make an error in making protein, it's not there forever. Proteins have a lifetime. They get broken down as well. Okay? Well, so the probability of synthesizing an error-free protein of 100 residues, 300 residues, or 1,000 residues, all right? If we look at the error frequency that we have, what's the likelihood we're going to make things of this size, all right? If we look at the error rate for making a protein of 100 amino acids, these error frequencies are pretty darn good. Even at 10 to the minus fourth, 1 in 10,000 errors, okay, we still have a 99% likelihood of making a 100 residue protein perfectly well. And even if we go over here to 1,000, we're still hitting 90% out here. Okay? So error frequencies for aminoacyl tRNA synthetases tend to be in this range. Okay? They tend to be in this range. This means that cells basically would have about 1% or less of their proteins that would have one amino acid that would have an error. Okay? That's not bad. That one amino acid might not even have any effect on the function of the protein. Okay. Boy, there's a mess. Now, it turns out that there are two classes of aminoacyl tRNA synthetases, and they actually do slightly different things. All right? Now, I want you to think about that CCA sequence that's at the 3' end of the of the tRNA. Okay? So we have a CCA at the 3' end. That's where the amino acid is going to get put onto there. That very last base has a ribose sticking out there. Okay? That ribose has a 2' hydroxyl and it has a 3' hydroxyl. Okay? And it turns out that the two classes of enzymes put the amino acid onto different hydroxyls. Class 1, which you see over here, and no, you don't need to know which ones are in which class. But class 1 puts the amino acid onto the 2' end of the A. Class 2, not surprisingly, puts the amino acid onto the 3' end of the A. Now, so that means that the amino acids are in slightly different positions depending upon whether they're on the 2' or the 3' end. It appears to have no significant effect on translational efficiency, but it is interesting to note that they come on in different ways. The two different enzymes also bind to and recognize things in slightly different ways. Class 1 looking like this, class 2 looking like this. And you'll look at this and you'll say, but wait, don't you have to look at the anti-codon on there? Okay? Well, the answer is they do. That's part of their binding in some cases. In other cases, it appears they don't make a lot of connection with the anti-codon. So the question then arises, well, how do they know which tRNA to put the right amino acid on? And it appears, and it's especially in this class here, it appears that there are other modified bases in the tRNA that differ between tRNAs. In other words, there may be another genetic code that's buried in here that tells this enzyme this is the right tRNA to put things on. That's kind of a cool thing. Okay? Kind of a cool thing. But in any event, the two different classes fundamentally put the amino acid on in different ways and in different places. Okay, I described a little bit last time about ribosomes, and I want to say a little bit more about ribosomes now. Okay? Ribosomes, of course, are the protein RNA complexes that make up the translational machinery. What you see in the figure is the actual structure, amazingly enough, of a prokaryotic ribosome. All right? As I mentioned last time, the ribosome has a large subunit and a small subunit. The large subunit is called the 50S, and in prokaryotes, that contains both the 23S ribosomal RNA and the 5S ribosomal RNA. Okay, 23S and 5S. Okay? The 30S subunit, also called the small subunit, this guy's called the large one, no surprise. The small subunit contains the 16S ribosomal RNA. Okay. Now, when we put these two together, we make a large, the complete ribosome, and the complete ribosome has 70S. You'll notice that the S units are not additive. 70 is larger than 30 or 50, but 70 is not equal to 30 plus 50. Don't ask. Well, there's reasons, but we're not going to talk about why. All right. So the prokaryotic ribosome is 70S. Now, when we look at a eukaryotic ribosome, we see a lot of similarity. All right. It too has a large subunit. It too has a small subunit. Okay. In the case of eukaryotes, the large subunit is 60S, and it contains 3 ribosomal RNAs. Okay. contains a 28S, which is equivalent to the 23S. It contains a 5.8S, which appears to be unique to the eukaryotes. And it contains a 5S. In eukaryotes, the small subunit has a size of 40S. So they're bigger. The eukaryotic ribosomes are bigger. And instead of a 16S, it contains an 18S, and yes, it is quite similar to the 16S of prokaryotes. The two subunits together make up the entire ribosome, and it has a size of 80S. So, now, I'm going to give you some functions for both the 16/18 and the 23/28S ribosomal RNAs as I get going through the lecture. Okay. All right. So when we go talking about translation, last time I mentioned that inosine is a base that frequently appears in the wobble position of tRNAs. Now, after I showed you the genetic code, I hope you recall that the wobble position of the tRNAs is one that doesn't matter an awful lot with respect to specifying an amino acid in the genetic code. And I mentioned that inosine turns out to have a very useful function in that respect. It turns out that inosine can form stable base pairs with 3 different bases. Okay? Here they are. Inosine with cytidine, inosine with uridine, inosine with adenosine. This one base can pair with all 3 of those. Well, what does that mean? It means, then, that the cell doesn't need to make 3 different RNAs, one to pair with cytidine, one to pair with uridine, and one to pair with adenosine in the wobble position. It only needs to make 1. So the cell, in theory, would make 64 tRNAs. It makes somewhat less than that. It doesn't need to make all those thanks to these possibilities raised by inosine. Okay? And there's the pairings, blah, blah, blah. Another thing that we see in RNA that we don't see in DNA is that GU base pairs are not unstable. It's another reason that U isn't a real good base for DNA. GU base pairs are not unstable. All right? So that means that we've got a lot of possibilities now that we can put in there for that 3rd position in the codon for the wobble position. Okay. Now, one of the things that we have to think about is, well, how does the process of translation go on? And in prokaryotes, the problem is actually kind of a complicated one. There's a couple of reasons. Now, if you recall in the genetic code, I said that AUG was the start codon. That was the thing that told the ribosome where to start translation. It turns out that AUG, when we look at a messenger RNA made by prokaryotes, all right, AUG, first of all, is not found at the 5' end of a messenger RNA. It's internal to that. So when we make a messenger RNA, when bacterial cells and us as well make a messenger RNA, they do not start with AUG. They start with something else. It goes in a ways, and then there's an AUG. Okay? It turns out that AUG is the only codon that codes for methionine. Methionine is found places besides the start codon. Methionine is found in the middle of proteins as well. Then to complicate it further, the first AUG that appears in the messenger RNA is not always the one that the cell wants to use to start translation. The upshot of all these things I'm telling you is the cell needs to have some other mechanism, at least bacterial cells need to have some other mechanism of identifying which AUG is the place to start making the protein. Which AUG is the place to start making the protein? Well, it turns out they have something. They have something called a Shine-Dalgarno sequence. And I think I have it on here somewhere. Where? All right, well, I'll spell it for you. Shine is one name, dash, Dalgarno, Dalgarno. I'm going to show you what that is in a second. Okay? I've lost my thing here. Accuracy. Where's my Shine-Dalgarno? Oh, here we go. Okay. Now, you recall when we examined promoters and we looked at the place where transcription started, we saw that about 10 base pairs ahead of the transcriptional start site that bacterial cells had a common sequence. We called a Tata box. All right? Well, if we look at the messenger RNAs and we look at the actual AUG that the cell uses, we discover that there's a common sequence about 6 to 10 base pairs ahead of that AUG. Now, that common sequence is called the Shine-Dalgarno sequence. And the consensus of that is GGAGG. This sequence right here is the core consensus. GGAGG, there's an AAGG, a UAAGG, GGAGG, etc. So GGAGG is the common theme that's in there. When we see a common theme that's conserved over evolutionary time, we recognize it must have a function, and the function that the Shine-Dalgarno sequence has is that it pairs with a sequence in the 16S ribosomal RNA. It forms a stable base pair with a sequence in the 16S ribosomal RNA. As we shall see, this base pairing between the Shine-Dalgarno sequence and the 16S ribosomal RNA helps to tell the ribosome which AUG to use. Here's an AUG that's not used. Here's an AUG that is used. Okay? So the Shine-Dalgarno sequence helps to tell the ribosome which AUG to start at. Okay, questions about that? Okay. Now, so that's kind of cool. Now, this turns out to be unique to bacteria. We'll see when I talk about eukaryotes that we don't have those, and we don't have as much of an issue with that, and you'll see why that occurs as well. But I'll talk about that in just a bit. One last thing I need to tell you before we start translation, okay, to actually go through the mechanics, is that prokaryotes have a fundamentally different setup for their genes than eukaryotes do. A very fundamentally different setup. Okay? Prokaryotes will not uncommonly have a collection of genes all that end up on the same messenger RNA. That is, they may have more than one gene on a given messenger RNA. This collection of genes is referred to as an operon, O-P-E-R-O-N. Eukaryotes do not have that. Eukaryotes have one messenger RNA, they've got one gene on there. Prokaryotes have multiple genes on a given messenger RNA. That we call an operon. Technically, the definition of an operon is a collection of genes all under the control of the same promoter. A collection of genes all under the control of the same promoter. Okay. Now, understanding that, yes?
Sorry, isn't the promoter part of the DNA sequence?
The promoter is part of the DNA sequence. That's correct.
So why do we call a collection of genes on an mRNA an operon as opposed to...
Okay, because the internal genes don't have separate promoters. There's only one promoter for the whole thing.
Right, but so the operon is the DNA part
The operon is the collection of genes. That's right.
on the DNA.
So they're on a DNA. That's right.
So it doesn't refer to the mRNA.
Oh, I see what you're saying. So let me get back to that. That's why I have a definition. I described what an operon is, but the definition is it's a collection of genes under the control of the same promoter. So from what you're saying, it is within the DNA that ends up in the messenger RNA. Does that make sense?
Sure.
Okay. That's why I gave you a description and then a definition. Okay, so a collection of genes under the control of the same promoter. All right? Now, that's important because it turns out that in prokaryotes, if they've got a messenger RNA and they've got a whole bunch of genes on there, they might have 10 genes on a given messenger RNA. You got 10 different places where you got start codons. You got to start here, you go out, you finish, and now you've got to find the next one and start and go along. So because there are multiple genes on a messenger RNA, having a Shine-Dalgarno sequence to tell the cell this is a start codon to use is a very, very important and useful thing. Okay, we'll see that as we get further into the mechanism. All right, let's look at the nuts and bolts about how translation proceeds. Translation is a very orchestrated phenomenon. I referred to the making of the splicing apparatus as a very orchestrated phenomenon. I'm not sure if I gave you guys the name. I put it in the highlights, but of the splicing apparatus, did I give you guys the name, the spliceosome? If I didn't, I should have. It's in the highlights. Spliceosome is the complex of all those proteins and RNAs that come together that do the splicing. It's called a spliceosome. A ribosome is a collection of proteins and RNA that come together to make protein. All right? Now, what we're seeing in these mechanistic things is, first of all, how this entire apparatus is assembled. So we start at the very beginning with the small ribosomal subunit. We're looking at prokaryotes here. 30S ribosomal subunit binds to some initiation factors. Initiation factors are proteins. And you're going to like the initiation factors. They're very simple. They have the names IF1, IF2, and IF3. Initiation factor 1, initiation factor 2, and initiation factor 3. We're not going to worry about the order in which they bind. Okay? These guys bind to a ribosomal subunit. The third initiation factor, which in this case is IF2, binds to the initiator tRNA. What's the initiator tRNA? Well, that's the one that's going to be complementary, okay, to the AUG in the messenger RNA, and it's going to have methionine on it. Okay? It's going to be complementary to the AUG in the messenger RNA. It's going to have methionine on it. You say, well, wait, what's the F on there mean? All right? Well, bacteria do a funky thing. They modify the very first amino acid that goes into the protein. They put a formyl group on it. And I won't talk about it here. I'll talk about it when I talk about eukaryotes. But they put a formyl group on here to protect it, basically. Okay? So the initiator tRNA will have an F-met. It has a sequence at the anti-codon that's complementary to the AUG. And it is brought to this assembling complex by IF2. IF2 is one that you should know the specific function of. Okay? IF2 carries the initiator tRNA to the complex that's being assembled. Now, what we will see is that IF2 does something kind of cool. And we'll see a related protein that does something very similar to it. IF2 comes and it grabs a hold of that initiator tRNA at the place where the amino acid is joined to the tRNA, effectively covering it up. Effectively covers it up. Okay? There's a reason why it covers it up, and I'll explain that in a little bit. At this point, we have 3 initiation factors. We have a small subunit, and we've brought in a messenger RNA. The other 2 initiation factors are helping to put the messenger RNA there. Since we're talking about the small ribosomal subunit, and the small ribosomal subunit has the 16S ribosomal RNA, not surprisingly, the Shine-Dalgarno sequence of the 16S ribosomal RNA over here is forming a base pair with the messenger RNA. Okay? I said that wrong. The Shine-Dalgarno sequence in the messenger RNA is forming a base pair with the 16S ribosomal RNA. And that's happening over here. Because of that, this AUG has been placed in the proper position on this 30S ribosome. So we've assembled the very first part, and we've got the messenger RNA in the right place for translation to begin when translation begins. Okay? Questions on that? This is kind of mechanistic. Yes?
So is that implying that all the Shine-Dalgarno sequences are a set distance away from the specific AUG?
Yes. So she says, does that imply that the Shine-Dalgarno sequences are a set distance away from the AUG? And the answer is yes, they are within a few nucleotides. So that's helping to position. This is the one that you want. Okay? All right. So we've made this initiation complex, and now basically these factors start going away. The IF1 and the IF3 go away. The large subunit comes in, and we form this sort of upside-down snowman structure that you see here. And at this point, we are ready. We're done with initiation. We have finished the initiation phase. To refresh everything here, we have a small subunit on the bottom. We have a large subunit up here. And as we will see, at this point, there are 3 different places now for transfer RNAs. 3 different places. They're called the A, the P, and the E. A, P, E, as we will see. This guy starts out in the P site. It's the only tRNA that starts in the P site. All of the other tRNAs come into the A site. Only the initiator starts in the P site. Excuse me. Okay, so we're now ready to really begin the process of translation, and that's going to happen in the next phase I will call elongation. So just like we saw initiation, elongation, and termination and transcription, so too do we see initiation, elongation, and termination in translation. All right, now, a little more closely, we can look at this and we can see, well, there's the A site, there's the P site, and there's the E site. They each have functions, as we shall see. At the moment, we only have one tRNA in there, and that one tRNA is the initiator. You'll notice that the initiator has its amino acid stuck up here in this tunnel that I described last time, because the polypeptide chain is going to go up through that tunnel as translation proceeds. Okay, now we've got another. Now we're ready to translate. So we've got a codon that's over here that's specifying what the next amino acid is to be translated. That codon is complementary to a specific tRNA. Okay? That means we've got to get the right tRNA in this complex in order for translation to proceed. Well, how does it get there? Well, this isn't very accurate, and this is showing you a scheme, but it's not very accurate in the sense that they're showing you the proper one getting there. They don't tell you how the proper one gets there. All right? Aminoacyl tRNAs come in bound to a very interesting protein called EF-TU. EF-TU. We call it EF2 is what we call it, although I emphasize it's TU is the way that it's spelled. Okay? EF2, all right? And by the way, all of the elongation factors in elongation are called EFs. Initiation factors are called IFs, all right? And the other ones, the elongation factors are called EFs. And there are 3 major ones also here. EF-TU carries the aminoacyl tRNA. And it carries it in a fashion very similar to what IF2 did. It binds it right there at the junction of these 2 things. And it turns out that's very important. The bond that holds the amino acid onto the tRNA is extremely sensitive to water. If water encounters that bond, it will break it within about a minute. It doesn't take much to break that bond. The reason that these proteins are covering up that amino acid at that point is they're precluding water from having access to that bond. Now, EF-TU, in fact, this is a very important thing. If we don't have enough EF-TU, what happens to the aminoacyl tRNAs that we just made? They're gone. They're broken down, right? EFTU turns out to be the most abundant protein in E. coli. Very important, okay? It's the most abundant protein that E. coli makes. Meaning that once they make an aminoacyl tRNA with the synthetase, they darn sure better have EFTU around as quickly as possible to cover up that bond and keep it from becoming broken. EFTU is a G protein. I didn't point out to you, but IF2 was also a G protein. You saw a little GTP bound to it. G proteins, you remember from last term, are small proteins that bind GTP, and they also cleave GTP. Slow down. Everybody got it? They also cleave GTP. All right. Now, how does EFTU function besides carrying things? It has another function. It brings the aminoacyl tRNA to the A site, and it asks the following question. Did I bring the right one? How can it tell? Well, if the base pairs form properly, it brought the right one. And if the base pairs form improperly, it did not bring the right one. Okay? If the base pairs form properly, it cleaves its GTP and exits. If it brought the right one, it cleaves its GTP and gets out of the way. On the other hand, if it brought the wrong one, it just backs out of the parking place and goes and looks for another one, carrying the tRNA with it. It doesn't want to leave the tRNA behind, of course, because that would mean it left the wrong tRNA there. Okay, so assuming we've got the right one there, we have a situation like this. We have the initiator tRNA with its amino acid there in the tunnel. We have the newly input tRNA in the A site, P site and A site. Next, the peptide bond is formed. Now, you remember the peptide bond is the bond, the covalent linkage between the adjacent amino acids. This turns out to be a pretty cool thing. The covalent bond is formed in the large subunit, kind of like you see here. What catalyzes the formation of the peptide bond? A ribozyme. Okay? The 23S ribosomal RNA catalyzes the formation of the peptide bond. It catalyzes the formation of the peptide bond. That means that all of the peptide bonds on the face of the earth are essentially made by a ribozyme. That's only been known for about 10 or 15 years. Okay? That's pretty cool. Well, this joining is made as follows. Okay? This guy here attacks the base and gets that attached to it so that we have the green, blue. Here was the green on the right, here was the blue. So the blue is the farthest one out. That's the amino terminus. And you'll notice that joining this bond results in kicking out the top part of this tRNA. We've sort of shifted everything partly by shifting like this. We next need to shift the bottom part, and that next shift of the bottom part involves a step called translocation. This translocation requires energy, and it requires GTP. By the way, you'll notice everything that happens in translation requires energy from GTP, not ATP. G proteins use it. And by the way, this elongation is catalyzed by a G protein called EFG. Okay? A G protein catalyzes the translocation, so now we're just sliding along. So what has happened at this point is the ribosome has moved one codon down. This first transfer RNA that was here is now sitting in the third site, which is called the E site. E stands for exit. And the primary function of the exit site is to kick it out. Now we're right back where we started. The only difference is now we have 2 amino acids on here instead of having one amino acid. We're now ready to go back up here and do the same process over and over and over. Okay? A lot of mechanics here. A lot of mechanics. Let me just summarize that. Okay. We finished the initiation complex. We have a transfer RNA in the P site. EFTU brings in the proper aminoacyl tRNA, cleaves as GTP, EFTU leaves, and it leaves behind the tRNA. You might say, well, what about that covering up of water, protecting it from water? Well, it has just placed it in the middle of this large subunit. There's no water. Okay, so this guy is protected from water just like this guy is protected from water. The peptide bond is formed by a result of catalytic action, the 23S ribosomal RNA. That results in a partial shift. The remaining shift is catalyzed by a G protein called EFG, which uses energy from GTP to slide this guy over. The original tRNA goes out the E site, and we're ready to go back and continue the process. Okay. Well, this is the worst part. Yes, sir? I'm sorry?
Does the peptide ever leave the P site, or does it get put on the A site transiently during the process?
Does the peptide ever leave the P site? Does it ever leave this? Is that what you're asking right here?
Yeah.
The peptide is always in that little tunnel. Yes.
Always.
It's always in that tunnel because remember, this is going to be snaking its way out of up here. Okay, so all the action as far as the peptide is concerned is happening right here. Everything else is just shuffling things out here for the tRNAs. Okay? Yes?
Does codon induce any kind of change that raises the affinity from one particular tRNA over another, or is it just guess and check that the EF2 puts in the right?
Say it again. I'm sorry.
So is it just by guess and check that EF2 brings the correct?
Oh, I see. Yes. Yeah. Is it random how it brings in the right one? That's basically your question. And as far as we know, yes. That's it, yeah. Now, what's remarkable is, of course, again, when we think of the nanoscopic level, which is where these things are occurring, we've seen that things can happen pretty darn fast there. We saw the carbonic anhydrase, which catalyzed 1 million things per second. So we know things can happen very fast at that diffusion level. This does happen rather fast. This is happening this has to support the rate of translation, which is about 15 to 20 amino acids per second. So that means that if it's completely random and we have a 1 in 20 chance of getting the right one in there, we have to have a lot of diffusion going on there, a lot of checking before we get the right one, and we move on. And we do that sufficient to support 20 of those or so a second. It's pretty remarkable to think about. But yeah, diffusion is pretty awesome. Okay. Well, I've got one more thing for us, and that is termination. Okay? Termination occurs when during this sliding process of one codon at a time, a stop codon appears in the A site. Remember, a stop codon is a UAA, a UAG, or a UGA. All right? When any of those appears in the A site, the ribosome sits here with nothing to do. There are no tRNAs that will form base pairs there. Okay? Instead, there are some termination factors called release factors that can come in and facilitate something very interesting. So in termination, here's a release factor called RF1. Release factor comes in and it binds at the A site because there's nothing else that'll bind with that UAA. The release factor comes in and it brings one thing to the ribosome. What do you suppose it brings to the ribosome? One molecule of water. That one molecule of water acts exactly where you would expect. It works right there, and the polypeptide gets released. Okay? As a result of that, there are other release factors called RRFs that will come along and facilitate the partial dismantling of this complex. It doesn't completely dismantle, but it is partially dismantled. Now we're almost out of time, and I think it's appropriate that we should pay some respect to the ribosome. Please stand. Please stand. Please place your hand on your heart and join me. Oh, beautiful with RNA to make the peptide bonds. You hold tRNA so it can pair up with codons. The ribosome translate mRNA, initiate and translocate from start to UGA. Thank you.